So we're going to start here with an examination of the system as a whole, and then we'll get into the individual components of the system as we move along here. So the first thing we're going to look at here is what are the organs and tissues that we'll see within the system. And so we'll start with what's usually referred to as the oral opening or the upper part of the digestive tract. This part itself is not part of the actual alimentary canal. This is going to include things like the tongue, the salivary glands, all of the various teeth. It ends at the glottis and epiglottis. That's going to separate the respiratory tract from the digestive tract in terms of anatomy. We will then get into the esophagus, and the esophagus will lead into the stomach. The stomach will then lead into the small intestines, and we'll get to each of the individual components of the small intestine here. We will then get into the large intestine. We will then get into, and the large intestine has different components and different structures within it, and we'll get into the individual components of the large intestine as well. We will then go into the rectum and then finally exit through the anus. This whole tube is an open tube through the body. Attached to these tubes is three organs that act as what we refer to as accessory organs. They are the liver and gallbladder and the pancreas. So we're going to ignore the individual components that are not part of the alimentary canal, and we'll focus a little bit here on the alimentary canal itself. So the alimentary canal itself, even though it's inside the body, is technically not inside the body. It's an open tube that runs from the oral opening to the anal opening that is set up in such a way so as to allow materials to be digested and absorbed without ever entering into the body until they've been absorbed. What this does is this creates a physical barrier, very similar to what we looked at in the immune system as relates to the physical barriers that we have preventing pathogenic and infectious agents from coming in. Within the alimentary canal, what we have to remember is that it's going to be about 8 meters in length from mouth to anus, independent of the individual's height. Now, that's for the adult. Children will have slightly smaller alimentary canals. The alimentary canal will grow as the individual grows, which means that shorter people will have slightly shorter alimentary canals and taller people will have slightly longer alimentary canals. But we need this length in order to allow for the distance and area necessary to have full digestion and absorption of all of the stuff that we are bringing into the body. We break it up into three parts. The three parts we'll reference as being the upper, the small intestinal tract, and then the large intestinal tract. So the upper tract is from the esophagus through the stomach. That's about a half meter in terms of the total distance that we have. We then have the small intestinal tract, which is made up of the three segments of the small intestine, which include the duodenum, the jejunum, and the ileum. That's going to be the bulk of this length of system. We will then get into the cecum, the large intestine, and the large intestine will be broken up into its individual components, which include the cecum, the ascending colon, the transverse colon, the descending colon, the sigmoid colon, and then we'll get into the rectum and then finally the anus. Each part of this tract has different anatomical structures that are going to match what their functionality happens to be for the overall function within the alimentary canal. Movement through the alimentary canal is regulated and controlled by peristaltic motion, which we'll talk about in a little while. But what's happening within the alimentary canal is we're constantly propelling foodstuffs, either in the form of a food bolus or as a chyme, constantly forward. And by forward, I mean going from the mouth to the anus. This motion is controlled by circular muscles, which we refer to as sphincters, and each sphincter is going to regulate the separations between structures. Some of these sphincters are dual sphincters, which means that they have an upper and a lower or an internal and external, whereas some of them only have a single sphincteral. The first such sphincter that we'll come in contact with is going to be at the glottis and the epiglottis. Now, this is not a true sphincter. However, it is necessary to ensure that swallowing takes place and peristalsis is initiated. And this is going to separate the oral opening from the esophagus itself. The next sphincter we get to is a dual sphincter. There's an upper and a lower sphincter to this. It's what's referred to as the esophageal sphincter or the esophageal gastric sphincter. This is going to be the separation between the stomach and the esophagus. There are two sphincters, and what these sphincters do is that they coordinate their relaxation and contraction so that both sphincters are not open simultaneously. This sphincter is going to regulate the rate of movement of materials from the esophagus into the stomach. And so if you can think about issues where that sphincter is not going to cooperate with the motion, we might have some sort of pathological conditions set up. The next sphincter we're going to get to is, once again, a dual sphincter. It has an internal and an external sphincter to this. This is the pyloric sphincter, and this is going to be separating the pyloric canal from the duodenum within the small intestine. And this one is going to allow for small pulses of what is now chyme, liquefied foodstuffs, to exit from the stomach into the duodenum. This coordinated function here is to prevent excessive amounts of materials heading into the duodenum, so as to first protect the duodenum from excessive acidification, and two, allow for proper digestion of materials once it starts to enter into the duodenum and into the small intestines. The next sphincter we come in contact with is what's referred to as the cecal sphincter, or the iliocecal sphincter. This is the separation between the ileum and the cecum of the ileum of the small intestine and the cecum of the large intestine. There's no real overall regulation, functional derivation between the cecum and the ileum in terms of what we're trying to control for. However, what it does do is it does provide a small little pause in between the two intestinal regions. The last of the sphincters is the anal sphincter, or the anus. Once again, we have two sphincters. The two sphincters here are going to be the internal and external sphincter. The internal sphincter is non-controlled. It's based off of pressure within the rectum, where if pressure within the rectum starts to increase, the sphincter will start to relax so as to reduce the amount of pressure within the rectum. The external sphincter is under cognitive control. This cognitive control is learned in the first 4 years of life, with mastery usually around the 5th year. And over time, as the nervous system starts to reduce its functionality, we slowly start to lose that overall ability to control the external sphincter, which can lead to issues, particularly for the geriatric population that we might be dealing with within our careers. When we talk about the regulation of the pathways here, it's about what's referred to as motility. And motility begins with the contractions of the circular and longitudinal muscles within the esophagus, following glutination, following swallowing. That will lead into the stomach. In the stomach, we have a slight difference in the coordinated contraction between the longitudinal and the circular muscles. That is going to slow down the rate of motility through the stomach so as to allow for full acidification of the materials that will lead to liquefaction into the chyme from the bolus. The rate of motility is going to be regulated based off of the materials that are being digested, the regularity that those materials are being seen, and any type of myenteric efferent activation through regulations by autonomic nervous system. Now, when we talk about the regulation based off of materials, what we're talking about is, does the secreting cells that are going to be producing the digestive enzymes produce those digestive enzymes? If they're not producing those digestive enzymes, those cells basically start to slow down their normal metabolic activity, which will reduce the rate at which those new nutrients are being digested. The more stuff we have within the alimentary canal, the faster motility will take place. The more materials that are there, the faster motility, and the less digestion will take place, leading to a larger volume of fecal matter at the end of the digestive process. So peristalsis is the coordinated actions that take place between the longitudinal and the circular muscles based off of the stretch reflex, either from the bolus or from the chyme of food as it enters into the lumen of the alimentary canal, in which the circular muscles will stretch, causing them to compress, doing a ring squeeze around the bolus, which causes a stretch in the longitudinal muscles, which causes a compression, which causes a stretch within the circular muscles. And so we get this squeeze, compress, squeeze, compress, squeeze, compress wave of motion within the muscularis layer of the alimentary canal that will constantly be propelling the food, the bolus or the chyme of materials being digested in a forward motion, meaning it's always attempting to move from the mouth to the anus. This begins with swallowing and terminates with the defecation reflex, leading to the elimination of fecal matter. Now, what ends up happening is that when we get to the stomach, we slow down this rate of peristaltic motion, allowing for increased activity and increased interaction between the digestive enzymes and solutions and the food materials itself. The motility that we see within the stomach is going to be referred to as gastric motility. The motility within the esophagus is referred to as esophageal motility. The motility within the intestines is referred to as intestinal motility. The rate at which motility is taking place is based off of the regularity at which we're seeing the food materials, and this regularity of food materials is going to dictate the rate at which contractions are taking place based off of the reflexive loop between the enteric submucosal plexus and the myenteric plexus. Within the stomach, what ends up happening is that there's a tertiary layer of muscle. And the tertiary layer of muscle is at an oblique angle to the longitudinal and the circular. And what ends up happening is that based off of the oblique layer firing, we end up getting this kind of circular action within the stomach. The circular action within the stomach leads to a regurgitation wave within the peristalsis. This regurgitation wave within the peristalsis leads to what's referred to as retroflow. There's a special anatomical region within the stomach, the fundic region, that allows for this retroflow to take place without having reflux into the esophagus due to the coordinated contraction of the sphincters during the peristalsis that doesn't allow for the bisphincter, esophageal sphincter to open when material is in that region. Now, regardless of what you've been told, the stomach itself will empty in terms of material in about a half hour, hour at most. The area of the fundic can hold material in it for about 20 minutes. So that gum that you swallowed when you were 7 years old is not still in your stomach regardless of what your mother told you. She simply didn't want you swallowing the gum. As material starts to be digested down, its density starts to change relative to the surrounding materials, and what happens is it slowly starts getting propelled towards the pyloric region of the stomach and then eventually out through the pyloric canal in the pyloric sphincter. And the pyloric sphincter is very narrow in diameter, and once again, just like with the esophageal sphincter, we have a bisphincteral layer, which means we have two sphincters that are going to be controlling the action at the pyloric sphincter, so as to control the rate of material moving into the duodenum so that we can neutralize the material at a fast enough rate so as to not cause etching of the mucosal layer within the duodenum of the small intestine. So what's going to regulate the rate at which stuff is moving through the stomach? So we have basically three things that are going to be regulating the material rate moving through the stomach, as well as the acidification of materials. Part of it has to do with how much is being consumed. The other part is that we're not actually going to be absorbing a whole bunch of material. Most of what is occurring within the stomach is liquefaction of the digested material. What can be absorbed is anything that is going to be membrane soluble. The most prominent thing that will be absorbed within the stomach are ethanols. But once again, most everything is going to be passed along in the chyme that's going to be exiting the stomach.